Okay, g'day. This is the last one. This is this strange new area. I suppose I'm partly responsible for being here. Um, and it's really about how do we use this knowledge? So what are we doing with science? Science is used to explain the world around us, but we also use the knowledge to change the world around us. And that raises all sorts of interesting questions, doesn't it? So let's have a look at some of these slides. I'm not too sure where to go with all of it, so hopefully I'll get it right and not wrong, but we'll find out. Let's pull myself away with there. So, gene cloning. To clone means to make an identical copy. So identical twins are in fact natural clones. The first attempts to clone were things like sheep, and Dolly the sheep's a famous sheep. And essentially, it shows us here we've taken a cell from this particular sheep. We've taken an egg cell from this particular sheep. We've taken the nucleus out of this sheep's udder cell and placed it and put it in the egg cell. So I've taken the nucleus out of this egg cell and replaced it. That cell fuses, it starts to develop into an embryo. The embryo is implanted into this foster mother, and the foster mother gives birth to a cloned sheep. Genetically speaking, if that was a 5-year-old sheep, that's a 5-year-old lamb. So genes are already starting to age, and Dolly did die young. Apparently, I can't think of the exact figures now, about 1 in every 100,000, 1 in every 1,000 of these works. Not a particularly productive manner of doing things. And clearly, as we get to understand the process, we're getting better and better at it. And you can clone a gene. So you can do a whole animal, or you can simply clone a gene. We talked about gene cloning essentially before. That idea of growing insulin in E. coli is essentially cloning a gene. So it has particular positive benefits. And it's not responding again. Come on. I'm in the way again. I'm going to put myself up there somewhere. DNA sequencing is obviously really important to all this, and so we talked at the last slideshow about gel electrophoresis and restriction enzymes and PCR and things, and we can use those techniques to in fact allow us to sequence a gene, sequence the whole genome. We've become very good at that, but also to sequence a gene. So you cut, using restriction enzymes, you cut up lots of pieces, and you end up with all these in your agar plate, and you start to put probes in and match probes with pieces, and you slowly build a map of where each nucleotide is. It's was quite a labour-intensive process. It was highly automated when the Genome Project got going, and we ended up with a lot of data being produced very, very quickly. So it cost a couple $100 million to do this first time. This is a part of the human genome. I think it's chromosome 1 from memory, or part of chromosome 1. But now with automation and ability to sequence these much more quickly, you can get your own sequence within a few weeks, and it costs less than $1,000. So it's becoming a really cheap and doable thing. So we can sequence each of your genes and see whether they've got mutations that are a problem. We can sequence the genome of species, and you do phylogenetic work. But it allows us, gives us a lot of information to do a lot of things. A lot of this is done using DNA probes. If you want to sort of, you put in a probe that has a particular known nucleotide sequence, and of course it'll attach to a piece of DNA with that sequence, or the complementary base pairs for that sequence, which allows you to do a lot of that work. So a lot of the DNA probes were used with the old Sanger hand method, and it's still the basis of a lot of the automated work as well. A lot of this sort of work originally was about paternity. I'm in the way, aren't I? Always in the way. So this is actually a gel that I ran many years ago. So you can see the gel, the wells here in the gel. This is where we loaded. So we loaded samples in this one, this one, this one, this one, and this one, but this lane was left empty. This here is a ladder. So these are known lengths. So each of the marks here is of a known number of nucleotides. each of these shows samples. So this particular lane had 2 samples of those 2 known lengths. This one 3, that one 3, and this one only 1 of a known length. So we could do that sort of work with them. So you can actually determine paternity. And as I've already got for paternity, I'll go down one. I've got a much better diagram in the next slide. Because there are sections of DNA that are non-coding, these non-coding areas often carry a great deal of mutations in them that are of no particular impact on the living creature, but of really interest because we can actually see the mutations within those. And family groupings have the same mutations. So this is a real gel. We look for what's called short tandem repeats, and these are bits of code between 2 and 5 bases long, or variable nucleotide tandem repeats, and these are longer than 5 bases, but not generally longer than 12. We don't want them terribly long. And look for these patterns in these non-coding areas. So you see you've got 1, 2, 3, 4, 5 ladders in place. As you see, there's quite a bend in there. So the multiple ladders allow for, well, that's probably a crappy gel really to work with, allow for some bending in the gel. But it is a fluid structure. And we go measure things across here. And then you've got these markers. Now, interesting enough, unrelated individual number 3 has 3 markers. These are normally alleles. There should only ever be 2. So individual 3 is an interesting character. Individual 4 is an interesting character. They're quite possibly carrying 3 chromosomes at that location. There's a control. Here's our husband. Here's his wife. She's going at 1, so she's homozygous. He's heterozygous. She's homozygous. The son clearly has an allele from dad and an allele from mum. The daughter, mum and dad. The grandfather, that's interesting. Grandfather's got alleles for both. And the grandmother is possibly related to the father, but not to the mother. He's possibly related to both, and that's a bit strange. So we can line these sort of things up and do some investigation. Clearly, one loci is not enough. This is just one loci, one locus, D4S139. And you do a number of these and build up a pattern. I keep moving. I'm going to rabbit on too long on this bit, aren't I? So, when we talk about GMO and transgenic, we're actually talking about the same thing. So, when a gene from an organism is purposely moved to improve or change another organism in a laboratory, the result is a genetically modified organism. That organism has had its genome changed and is therefore transgenic. So, it's a way of talking about the same thing. Transgenics have been used already for a number of interesting things. I'll get this out of the way again. Keep getting in the way. So a lot of food, fruit and vegetables particularly, and products made from them, you can use transgenics to actually improve how long they last on the shelf. Clearly, being able to put pesticide resistance into a crop, so you can spray the whole field for weeds and only kill the weeds, not the crop, is a useful thing until the crop plant leaves your farm and starts becoming a weed elsewhere and you don't have a spray to kill it. Or you can give the plant resistance to pests and therefore you don't need to spray as much, which is probably a better outcome. We've also used crop improvement to improve nutritional levels. There's a famous one where bananas were used to deliver cholera vaccines. Really useful idea. And obviously we can now make higher milk yield for dairy cattle, better meat yield on meat-bearing animals, sheep, goat, cattle, whatever. And we've also used transgenics in medical procedures and vaccine production. And indeed, we've actually made transgenic bacteria which destroy chemical spills and oil spills. So there's some really valuable things that can come out of transgenics. Here we go again. Change slide. But there are a number of issues. And we'll talk about issues much more in class, so we need to really tease these out. But essentially they're biological, social, or ethical. Now, if we are changing another organism genetically, we are causing it to evolve in a certain direction that we would like. And we've done that for a long time through crossbreeding, as this selective breeding at a faster pace. But we have the ability to reduce quite markedly the diversity within that species. And a species who lacks genetic diversity is at more risk within the environment of disappearing. And we're having a few of those problems already in some of our food crops. And again, I'll talk about that in class. I'm going to run out of time here. Socially, there's some issues around the social justice. If white elites in the Western world have control over this and get to choose when it's used and get to choose where they use it based on who can afford to pay for it, then a lot of these wonderful advances are great for those in the elite Western world. But where their needs are greatest in the majority world, amongst poor people who aren't white, we have a real issue. And there's some issues around that, which we'll talk about in class too. And then ethically, who gets to play God? A lot of my friends donated sperm in the 80s as university students. We weren't paid much for it, but they got $20 or something, got some beer money. But they did that on the understanding that any progeny, any children who happen to come from that sperm would not be able to find them. But we now have people who are seeking their biological fathers. And sometimes for very good reasons. Are you carrying a genetic trait that I need to know about before I have my own children because I don't want to pass on a disease? Who's right? Is the father who gave the sperm, took the money for the sperm to help research or whatever, should he be anonymous? Does the child who needs to know about potential change problems need to know what they could be? So there's some really interesting ethical questions around GMOs and transgenics. This is a wide-ranging session. I'm thinking in the future of hiding this in other places. What do we do with the disease? These diseases are crossing the species barrier into the human populations quite a bit. So what does the World Health Organization and other centers do? You identify the pathogen, then you identify the natural host of the pathogen and look for some solutions there. Establish quarantine, so you stop the spread, map the spread and its mortality, develop strategies for the next time it appears, and of course, more importantly, share all the information and start to develop treatments and vaccines. Again, we'll unpack a lot of that in class because there's a lot going on there too. You hear about pandemics and epidemics. Pandemic is simply an epidemic that's gone global, essentially. It's gone really big. It might be the whole country. So an epidemic could be an epidemic of flu in Melbourne this winter, or a pandemic of flu across the whole continent. So that's basically the difference between the two. Rational drug design. Oh God, we're going around time again. We've been able to make drugs to do certain things. Relenza is one of these wonderful ideas that came out of the late 80s, early 90s from CSIRO. And basically, it blocks a particular enzyme, neuraminidase, that allows the virus to leave the host. So if the virus can't leave the host, then it can't spread. So it's a really clever way of targeting the process by which a virus attacks the body. So we see a lot of rational drug design in the production of antivirals. So antibiotics used against living things that we discussed way back, basically from botanical sources and other sources, biological sources. But due to our overuse, their potency is really reduced because our overuse has acted as a selective pressure and they've learnt to adapt. Antivirals are used essentially against viruses and non-living pathogens. And these are things we are manufacturing for ourselves. So influenza, HIV, hepatitis, herpes, all being worked on. And most importantly, things like Ebola is now currently being worked on and with some great success. And I've run out of time, so I'll have to pack this out in class a bit more. But we've got plenty of time before the exam, haven't we? Not really. Okay, see you in class.